In progress. Okay, just a little of introduction about our speaker today. So Dr. Jon Stoessl is a professor and head of neurology and co-director of the Center for Brain Health at the UBC and Vancouver Coastal Health. He holds a Canada Research Chair in Parkinson's, served until recently as the director of Pacific Parkinson Research Center at UBC. And he has directed a CIHR team in Parkinson. Dr. Stoessl is a deputy director, editor of the Movement Disorder Journal and sits on the editorial boards of various medical journals, including Lancet Neurology. He has served in numerous scientific advisory boards and is the past chair of the Scientific Advisory Board of the Parkinson Society of Canada and Ontario Mental Health Foundation. He has served on various international boards and committees, co-chairing the steering committee for the World Parkinson Congress and is the vice president of the World Parkinson Coalition. He's a member of the Order of Canada and fellow of the Canadian Academy of Health Sciences. Dr. Stoessl's research involves the use of positron emission tomography to study Parkinson's disease and related disorder, including the imaging as a biomarker. He has more than 9,800 citation and has published more than 270 papers and book chapters. He has served as the medical director for PSBC board for a number of years. We are very fortunate to have him as a speaker today. Please join me in welcoming Dr. Stoessl. Over to you, Dr. Stoessl.
Okay, thank you very much. Just while I'm bringing the slides up, I'll just say that sounds like a very dated summary of me, because I've stepped down as head of neurology and as recently Dr. McEwen has taken over in that role, and I stepped down several years ago as director of the Center for Brain Health. And I'm now actually the editor-in-chief of Movement Disorders. So I'm going to talk about neuroimaging as a biomarker, as well as other fluid biomarkers in Parkinson's, but the focus will be on imaging. I have some disclosures which are not really relevant to this group, but they are, in as much as I do periodically get asked to advise various companies on, particularly on imaging biomarkers as outcome measures in clinical trials. And that's quite relevant. It doesn't have any impact on what I have to say to you today, but it is maybe relevant to some of the questions. So the role of biomarkers in Parkinson's, first of all, and one that I'm not going to spend a lot of time on, is diagnosis. Mostly the diagnosis of Parkinson's is going to be made, of course, on clinical grounds. And I don't request imaging in the overwhelming majority of patients who I see, because I'm going to make the diagnosis based on history and examination. But sometimes one can't be sure. And so imaging or other biomarkers may be helpful. And that may be particularly the case for early stage clinical trials, because if you enter somebody who has an atypical form of Parkinson's into the clinical trial, that could, of course, confound the results. And increasingly, although we don't yet have something approved that is known to slow disease progression, we don't have a pharmaceutical product approved that's known to do that. But there's a lot of active work in that area, and the emphasis is increasingly going to be on the ability to detect disease in early stages, or even before it becomes manifest in people who are at high risk. Once you actually start a clinical trial, you need some means to assess disease progression. And again, in the clinic, we usually do this simply based on examining the person. But if you really want to detect an impact of a novel therapy, you probably need better approaches to do that. And so imaging has historically been the best way to do that. And it probably still is, but it's very imperfect, as you will see. If you want to develop a new approach to treating disease, you need to know that the treatment actually gets into the brain and acts where it's supposed to. No point in having a powerful antibody if it doesn't actually cross the blood-brain barrier. So imaging could theoretically be very useful for that, to demonstrate target engagement. I'm not going to speak a lot about that, but we can discuss it later in questions. And then the whole approach to personalized therapy. Can you select people with particular characteristics or stratify them? In other words, give priority to different categories of disease for participation in clinical trials and for both clinical trial and clinic use. Is there a better way to predict how the disease is going to unfold in the individual? Can you predict who's going to be more prone to certain complications, who's less likely to do well with a certain type of medication, etc.? So these are emerging areas of interest that I will discuss. But one thing I really want to emphasize is that regardless of whether you're looking at imaging or a fluid biomarker, in other words, a test done on the blood or on the spinal fluid, there's no one size fits all. So the biomarker that you need depends on the particular question that you're asking. And people often forget that. So I'm not going to talk about all the standard imaging that is used. You're all aware that we will sometimes get MRI as a diagnostic test. And historically, routine MRI has not been regarded as all that useful for the assessment of Parkinson's disease. It's usually more helpful for excluding other Parkinson-like conditions. However, with newer techniques in MRI, we're going to see a lot of change already and over the next few years. So one approach is to the ability to look at neuromelanin. Melanin is the pigment that gives our skin color, but melanin is also produced as a byproduct of dopamine synthesis in the brain. And so there is neuromelanin in the substantia nigra, the so-called black substance. That's actually how it gets its name. And that's produced in the dopamine nerve cells that degenerate in Parkinson's. Neuromelanin is also seen in an area of the brain that produces the neurotransmitter noradrenaline. And that region of the brain is known as the locus ceruleus, the blue place. So you can detect changes in neuromelanin in Parkinson's, and you can see here, this is early Parkinson's. There's a decline in neuromelanin in the substantia nigra compared to healthy individuals. And then if you look at people with more advanced disease, that decline is more obvious, but it still changes over time. The problem is, in terms of diagnosis, this may help you detect the loss of dopamine neurons, but it really doesn't easily allow the differentiation between Parkinson's disease and the commonest forms of atypical Parkinsonism, which would be progressive supranuclear palsy and multiple system atrophy. Now, there are some subtle differences, but I'm really not going to dwell on those here. Another really interesting approach that is not considered routine or standard at this point, but I think is well worth keeping an eye on, is the ability to use MRI to detect water in the brain. This is free water, so it's not being constrained by the usual boundaries because the nerve cells die off. And so you can see that in early Parkinson's, there's an increase in free water in the posterior substantia nigra. And the degree of free water actually changes over time. So this may be a reasonable marker of disease progression. Not going to be perfect, and as I already indicated, it is not yet regarded as standard, and it requires fairly specialized expertise. This is work done by my colleague David Valancourt at the University of Florida in Gainesville, but he has also worked towards trying to make this technique more routinely accessible. So these people have published an automated approach to studying free water in the brain, and they can not only detect Parkinson's versus health, but they can actually differentiate between Parkinson's and atypical Parkinson's, and between the two major forms of atypical Parkinson's, multiple system atrophy and progressive supranuclear palsy, because the patterns of where the free water can be measured are different between these different conditions because the pathology is different. But the imaging method that everybody is most familiar with, although it's not widely available, and is the one to which I've devoted a great deal of my career, is the ability to image dopamine in the brain. So these are PET scans that were performed at UBC using 3 different approaches to study the dopamine neuron, which is shown up here in a cartoon form. So fluorodopa is basically levodopa, like Cinemet, only it's been labeled with a small amount of radioactivity. And when levodopa is taken up, it's converted to dopamine, which is then packaged in these membrane-bound sacs that can be released in the normal brain. They're released in a controlled fashion into the space between nerve cells, known as the synapse. And then when dopamine is released into the synapse, its action is terminated by taking it up again into the nerve cell that released it using the dopamine transporter. So we have 3 different ways of imaging dopamine shown here. One is fluorodopa, which is like levodopa. The other is a molecule that is responsible for packaging dopamine into these vesicles. So for that, we use a radio-labeled form of tetrabenazine. And then once dopamine is released, it's taken back up again through the membrane transporter. And there are a whole host of drugs that can be radio-labeled to image the dopamine transporter. We use methylphenidate, which is better known to most of you as Ritalin, used as a treatment for attention deficit disorder. So you can see here 3 scans from the same healthy individual up top. And then if you look at an individual with early Parkinson's, you can see that all 3 scans show a somewhat similar pattern. They're not identical, in which the posterior or back part of the striatum is affected first, and the 2 sides of the brain are not affected equally. So that pattern is characteristic of Parkinson's, and we can measure the degree of abnormality. But the problem is, this is not specific for Parkinson's, and you could see similar changes in atypical forms of Parkinsonism. So there are other techniques that can be used to try and differentiate between Parkinson's and these other atypical forms. This is a technique that was developed by my colleague David Eidelberg in New York, where they look at patterns of glucose use in the brain. Glucose is the main source of energy for the body in general, and in particular for the brain. And what you can see here is that in people with Parkinson's, there are relative increases in glucose use in the basal ganglia, paired with relative reductions in glucose use in areas of cortex, here the premotor cortex and the posterior parietal cortex. Now, that is quite characteristic of Parkinson's, and I will come back to it later, but what I want to show you right now is that if you look at atypical forms of Parkinson's, multiple system atrophy can be very difficult to distinguish from Parkinson's disease, particularly in early stages. But here you can see a very different pattern where the basal ganglia, hot in Parkinson's, cold here. And in fact, the same would have been true for the cerebellum, which is cold in multiple system atrophy, is hot in Parkinson's. Progressive supranuclear palsy has a different pattern. And so you can actually use these patterns of glucose use to differentiate. In Canada, we don't routinely use glucose PET for diagnostic purposes, except for cancer. And then you're usually looking elsewhere in the body, or sometimes for epilepsy. So this is not in routine use. The other thing that we often forget about and is not used very much in North America, but has become popular in other parts of the world, is to look not at the brain, but at the heart. Because in people with Parkinson's disease, the autonomic nerve fibers, the sympathetic nerve fibers that control the heart, degenerate. And so if you look here, this is the liver. This is where the heart's supposed to be, and you don't see anything here. In contrast, somebody with multiple system atrophy, where they also have loss of autonomic function, but where it is in the brain rather than in the periphery. So here you see preservation of cardiac innervation. So this is a useful way to differentiate between the two, but overall the differentiating capacity is not quite as good as it is with glucose PET that I just showed you a moment ago. So as I indicated earlier, there is a huge interest in the possibility that you might be able to detect disease before people actually show signs of Parkinson's. So in general, there are three groups of people who are at high risk for developing Parkinson's. One are people who have a reduction in the sense of smell. Those of you who have Parkinson's are already aware that you may well have lost your sense of smell and that may have occurred some years before you developed Parkinson's. So this is one study comparing people who have poor sense of smell, normal dopamine, somewhere in between, and abnormal dopamine. And you can see that the people with the abnormal scan have a much higher risk of going on to develop Parkinson's within a relatively limited time frame. The other group of people who are going to be at high risk of developing Parkinson's before they show clinical signs are people who carry a genetic variant that is known to be associated with Parkinson's. This is work we did. This was actually done with support from Parkinson's Canada to Daryl Weil, who's now practicing in Kelowna. And here we're looking at people who have a known genetic variant in the gene called LRRK2. And if you compare the healthy against somebody who's affected, that's an obvious difference. But you can see there's also quite a striking abnormality here in the person who carries the genetic variant but is not yet clinically affected. And we can look at the time course of that and look at different tracers. I'm not going to dwell on the details of that right now. The third group, and the one that probably attracts the greatest interest these days, are people who have so-called REM sleep behavior disorder. So REM sleep is when we dream, and normally when we dream, we're paralyzed to protect us against what we could do to ourselves, because often in our dreams, we're we're chasing criminals or we're running away from wild animals. In people with REM behavior disorder, the switch that leads to paralysis during that stage of sleep, the switch doesn't work. And so people are actually quite mobile then and may act out their dreams. And a number of you may be very familiar with that. So people with Parkinson's have REM behavior disorder, but so too, if you have REM behavior disorder and you don't have Parkinson's yet, your chances are increased. And so this looks at dopamine innervation. Sorry, no. In people who have RBD versus those who do not. And you can see that some people go on to develop, if they have an abnormal scan, they're more likely to develop Parkinson's. This goes back to the glucose pattern that I showed you before, the pattern that you see in Parkinson's. Well, some people with REM behavior disorder show this abnormal pattern, and they're the ones who go on to develop Parkinson's. And then a group in Denmark has done a really massive study here, looking not only at dopamine innervation up here, but looking throughout the body. So they've looked at the gut, cholinergic innervation in the gut, the heart that I showed you earlier, gone here. This is the noradrenaline innervation that I talked about earlier in the locus ceruleus, gone in the people with REM behavior disorder. And this is using molecular imaging to look at noradrenaline innervation in the brain. And you can see that it's been affected here as well in people with REM behavior disorder. So they're using this to try and study the pattern of progression through the body and up through the brain before disease actually becomes manifest. I talked before about free water imaging in Parkinson's, and you can see it's abnormal here, Parkinson's versus healthy, but if you look at people with REM behavior disorder, they're somewhere in between. And interestingly, if you look at people who have genetic variants, the same thing as before. This is people with a genetic variant compared to healthy controls, and people with a different genetic variant, which is the glucocerebrosidase variant, known to be associated with Parkinson's in some people. And you can see these changes emerging over time before disease actually becomes manifest. I'm not sure if somebody's trying to put something in the chat for me. I'm a little bit hesitant to try to. Oh, yeah, let's come back to that. Yeah, let's come back to that later. It's a good question, an important question. So, in fact, the question was, can you do anything? If you know that you're at risk, can you do anything to prevent it? And that's really where this next section becomes highly relevant, is how do you measure the change over time if you have a new treatment? So this is again work that we did looking at progression of changes in dopaminergic markers in the brain. And you can see that if you extrapolate back, we may be able to detect changes around 15 years before disease actually develops. But the other thing that I want to show you is that the pattern of change here is very similar to the pattern of change that is identified in post-mortem studies, although here they seem to level off at around 4 or 5 years. We may not show leveling off until slightly later, but it's overall very similar. We usually use dopamine markers to assess change, or other centers do. It's still considered the standard in clinical trials, but that may not necessarily be the best approach, because I showed you, one of the first slides I showed you was the changes in melanin in the brain. And in people with early disease, melanin is not a very sensitive marker. doesn't correlate very well with clinical change. In early disease, it's dopaminergic imaging that is probably more sensitive to detect change. But in people with later disease, the neuromelanin may become more useful. We have problems in that we assume that the dopamine changes we measure in the brain correlate with cell counts, and that seems to be what's shown here. But I just need to tell you that that is controversial, and not all studies demonstrates that. So this is from a center that does very good work, and they find no relationship between dopamine transporter SPECT scans and the number of dopaminergic cells measured at postmortem. But the real problem is that even if you can reliably detect change, it doesn't necessarily translate into clinically important change. So this is work that we were involved in. The scans were performed at UBC, but it's from many years ago from fetal transplant studies. And you can see that from baseline to 2 years after the transplant, there's a marked improvement in the scan, but that did not translate into clinical improvement, unfortunately. This is from another study that we were involved in where we actually did the analysis of scans that were performed in England. This is looking at a growth factor, GDNF, and it may not show super well on the slides here, but this is from baseline to follow up in the placebo group. This is from baseline to follow up in the active group, the group that received GDNF. There's a marked improvement in the scan here. They come up to almost normal levels, but again, the study did not meet its primary clinical endpoint. And just a very brief word on target engagement, because molecular imaging can be very useful to demonstrate whether the drug that you're giving is actually affecting the brain. This is not from Parkinson's. This is the example of Alzheimer's disease. This is aducanumab, which is one of the antibodies that received a lot of press recently because its approval by the FDA was considered so controversial. Well, you can see that there's a marked impact, which is dose related. As you increase the dose, you clear out more amyloid from the brain, but it's quite controversial as to whether this translates into clinical improvement. And this is a summary slide looking at all the different, well, the slide is now a little bit old, so there are a couple of new drugs on here, but it's looking at a whole host of these similar drugs where target engagement was seen. That's the green boxes here. But in many cases, people either did not improve or may even have got worse. And in the case of aducanumab, there were 2 large phase 3 studies conducted, and they actually came up with different results. So the one study found benefit, the other one did not. And it simply shows you that you need to be a little bit wary in terms of interpreting all of this stuff. It's also being suggested that you can use DAT scan to, so this is dopamine transporter SPECT, to select people for inclusion in a trial. And although in general that makes sense, you see greater progression in the people who have the abnormal scan, it's important to remember that even people who have abnormal scans may deteriorate over time. And in a recent, very important study that I'm going to come back to later, the alpha-synuclein screening, the biochemical test, was positive in 9% of people who had normal scans, which doesn't sound like a lot, but that's the same as you would see in people who carry a genetic variant, and is more than twice as much as was seen in healthy controls. So it's not a perfect separator. There are other things, of course, happening in the brain in people with Parkinson's. It's not just the loss of dopamine cells. So there may be inflammation in the brain, and that may be a very useful target for therapies in the future. The problem is right now, I think in the interests of time, I'm just going to skip through this quickly. Most of the tracers that we have are not very reliable, although there are some recently that show differences, and the differences seem to correlate with peripheral markers of inflammation, in other words, blood test markers. This is one that just got published a few weeks ago. In fact, it's not even being peer reviewed yet. It's on a preprint server. And you can see this is a very different approach to studying inflammation in the brain. These authors have demonstrated inflammation in the brains of people with Parkinson's using imaging, but they're only seeing these changes in people with relatively advanced disease, not in early stages. So the meaning of this is actually not that clear. I'm going to skip over this one, and so that we can get to a discussion of alpha-synuclein. So this is the protein that is misfolded and is abnormal in Parkinson's disease. And in an ideal, so that's similar to amyloid or tau in Alzheimer's disease. And so if you want to study Parkinson's, the obvious thing is to develop a tracer that will label alpha-synuclein. Maybe I won't get into the details right now in the interests of time, but it turns out that this has been incredibly difficult. And I'm going to show you some extremely recent papers that have just appeared in the last couple of weeks. But one of the problems is that alpha-synuclein is not highly expressed in the brain. So it's harder to label it. The other thing is that all of the proteins that are misfolded in neurodegeneration, so in Alzheimer's disease, in ALS, take on this abnormal conformation of a so-called beta folded sheet. And that structural similarity, plus the fact that all of these proteins tend to be co-deposited in the same parts of the brain, means that you need an extremely selective drug to label alpha-synuclein, or you're going to be picking up something else. So we thought we had something promising here. This image actually got the cover of the Movement Disorders Journal, and you can see that there's abnormal uptake here, but it's in people with multiple system atrophy, either the Parkinson variant or the cerebellar variant. And in the cerebellar variant, you see abnormal uptake in the cerebellum. You also see it in the Parkinson variant, as you would expect. The problem is this tracer doesn't seem to show much binding in people with Parkinson's disease. So this paper just appeared earlier this year. And I'm not going to go through the details, but it does look promising. This is a new compound that seems to be binding to alpha-synuclein, and it appears to be selective. So it binds much better to alpha-synuclein than it does to amyloid or tau, which are seen in Alzheimer's disease. And you can see here, although these look similar, if you look at the scale here, the binding is much lower in Alzheimer's than it is in Parkinson's. They've done some preliminary studies in non-human primates using two different models, and this is the problem. You can see they have some binding here, but it's a bit messy. The signal is low, so it's not clear how useful this will be. And then this paper just appeared literally within the last few days. It's a different tracer for alpha-synuclein. It's one that's been heavily publicized for quite a long time now. But you see, again, just like the others that I showed you, much more convincing binding in multiple system atrophy than in Parkinson's disease, where you may see a little bit here, but nowhere near as much as in the basal ganglia and multiple system atrophy. So we're still not there. Now, this is work we did using a tracer that was actually developed for tau, so a different protein in the brain. But what we found was not only that it binds in progressive supranuclear palsy, which is where we would expect to see it, but we found it in people who have a genetic variant that leads to Parkinson's. But these people were studied when they were clinically normal. So we think it's binding to alpha-synuclein, and that's somewhat supported by post-mortem studies. The real question is, what can we learn about Parkinson's from doing these studies? So everybody focuses on the substantia nigra, which is where the dopamine cells are. But if you actually look at where the action is happening, alpha-synuclein mainly works in the nerve terminal, which is a long way away from the substantia nigra. And my colleague, Jose Obeso in Spain, has suggested that the problem really isn't originally in the substantia nigra at all, but rather in the projections to the striatum, to the basal ganglia. So these are the dopamine cells that project to the striatum. They're the ones that degenerate in Parkinson's. But the striatum gets a lot of its important input from the cortex. And that input is so-called somatotopic. In other words, it separates out different regions of the body, whereas the substantia nigra does not have that kind of representation. And we know that Parkinson's tends to be selective for certain parts of the body. And so his hypothesis is that depending on how you use the brain, that may lead to selective loss. And so this is work done by Michele Matarazzo when he was with us, but he's now returned to Spain, showing that the changes, and we think this reflects alpha-synuclein, are not in the substantia nigra so much, but rather in the basal ganglia, in the putamen here, which is the region of the brain that's most affected. And so historically, we've probably been looking in the wrong place simply because it was easier to do so. I'm just a little bit concerned about time here. So I just want to very briefly comment that there are ways of predicting the trajectory of Parkinson's. So this is one, which is to look at the degree of atrophy in the brain. I'm not going to talk about it in detail. You can also look at other chemicals, and that's why I said at the beginning that the biomarker you want to use depends on the question you want to answer. So we tend to focus on dopamine, but for people with Parkinson's, cognitive impairment or unsteadiness on the feet, loss of balance and falls would be more important probably. And those tend to be linked to cholinergic function in the brain. So you can look at that. This is work we did on the right here. Looking at people with genetic variants, and we actually found that they had preserved or increased cholinergic function, which fits with the fact that this particular genetic variant is associated with a lower risk of cognitive impairment. You can also do structural imaging to look at the cholinergic regions of the brain. That's shown here. And what I want to emphasize is this doesn't just correlate with cognitive deficits and problems with gait, but it actually predicts the people who will have future trouble. That's shown here for gait. And then you can look at changes in connectivity between different regions of the brain, and those may predict the change in motor function over time. But fascinating to me, they may also predict who is at risk for impulse control disorders in response to dopamine agonist medication like pramipexole. So these are people who were normal when they were studied, and the people who have this pattern of connectivity, the details don't matter, are the ones who go on to develop impulse control disorder after they're started on medication. I'm just going to skip that and cover, if I have time, just a few really important recent findings. So I've talked mainly about imaging, but there's a lot of interest in fluid markers. This is a protein called neurofilament light, which can distinguish between atypical forms of Parkinson's and Parkinson's, although it becomes slightly abnormal in PD. Not terribly good as a marker of disease progression, certainly not in Parkinson's disease. But the one that's really had a lot of attention, and you may well have read about it or heard about it on social media, is the ability to measure alpha-synuclein, abnormal alpha-synuclein, using a so-called seeding amplification assay. Now, the idea here is that we all have alpha-synuclein, but when it becomes misfolded, it can then trigger misfolding in the normally conformed protein. So if you take a well, you put normal alpha-synuclein in the well, and then you take your sample from the patient. If it's misfolded, if you wait long enough, it will cause misfolding in what was originally the normal alpha-synuclein. And you can measure that using fluorescence. And then as over time, the increase in fluorescence is indicative of the presence or absence of abnormal alpha-synuclein. And I want to emphasize right now, it's just the presence or absence, so it doesn't correlate with disease severity. This is the study that appeared in the spring that attracted a lot of attention. This was sponsored by the Micheal J. Foundation, the PPMI. And I won't go through all the details here, but you can see that if you look at this so-called seeding amplification assay, the sensitivity is high. In other words, most people with Parkinson's have it, particularly those who have an abnormal sense of smell. Not so high in the people who have the LRRK2 mutation that I showed you earlier. But even in those people, there's a difference between those who have an abnormal sense of smell and those in whom the sense of smell is normal. It's not so clear what happens in atypical Parkinson's, though. The signal is abnormal in multiple system atrophy, but at a much lower amplitude and probably with different time course. And so, again, if you compare the different forms of Parkinson's, the sensitivity for Parkinson's is quite high. The sensitivity for other synuclein disorders like multiple system atrophy is high, but sorry about this, the specificity is not very good. We can do better than this in clinic, at least with prolonged observation. So this is still not the ideal test, even though it's attracted a lot of attention. And there have been a couple of other recent developments. This is very similar work that was done in Japan, where they used a somewhat different technique that I'm not going to spend time on. And what they found was that the appearance under the electron microscope, comparing Parkinson's against other forms of synucleinopathy, was different, and you can actually separate between them. So here's Parkinson's, which has one particular pattern, as opposed to multiple system atrophy, where you see different types of pattern. So this is a very hot and emerging area. And you can actually even detect these proteins in the skin, because the skin has nerve fibers. This is alpha-synuclein, and there's a difference between Parkinson's, where the alpha-synuclein is around autonomic nerve fibers, as opposed to multiple system atrophy, where they're in sensory nerve fibers. And quite recently, people have done the same thing with the protein tau. I really don't need to spend time on that, and we'll just summarize. So just to reiterate, structural MRI is mainly useful for differentiating PD from atypical Parkinsonism, but some of the emerging techniques with MRI, like neuromelanin or the water imaging, may be useful as diagnostic and progression markers. Dopamine imaging is still the gold standard, despite the caveats that I told you about, but It's not as useful as a diagnostic marker. It is useful for assessing disease progression. We talked about glucose PET. And then finally, we talked about the fluid biomarker, particularly the seeding amplification assay. You're going to hear a lot more about this over the coming years. It's actually already available commercially in the US, fortunately not in Canada. And I say fortunately not because I think one of the concerns that we all have is, what do you tell somebody who feels perfectly healthy, who has an abnormal seeding amplification assay? Can you tell them that they are definitely going to get Parkinson's? I don't think you can at this point. So a lot of ethical considerations as well. I'm going to close there, but I'd be happy to take questions. Oh, there was a question earlier about if somebody has the signs of Parkinson's, of possible Parkinson's, like loss of smell, is there anything they can do before it manifests? That's a great question, and it links very much to my last comment about the seeding amplification assay. The question that people very reasonably pose is, what's the point of having all these tests or knowing the results if you can't do anything about it? And I would say, fair point, I agree, except that you can't do anything about it right now with pharmacological approaches, with drugs or antibodies. But there are things you can do because you can exercise, you can have a healthy diet. And you may think that's trivial, that it's kind of minimizing it. It's not. From everything we're learning, we believe that exercise has a profound impact, and diet may also have an impact. I'm sure you've heard my colleague, Dr. Cresswell, talk about the MIND diet and the Mediterranean diet. Question here about getting genetic testing. So that's a complicated, and two questions about genetic testing. So first of all, mostly this is done for diagnostic purposes, and it's actually not as straightforward as we would like. Right now, the only way one can get it done theoretically is through a commercial lab, and so there is a cost associated with it. And I don't generally request it. We've historically done this for research purposes. One of the other really important things is the ethical consideration of how you handle the results. So first off, it would be deemed unethical to do genetic testing on somebody who's underage, who is unable to consent to it. But we think that the most appropriate thing is, if you have somebody who's at risk, supposing you've got Parkinson's, it's thought to run in the family, so you may have an increased genetic risk. The appropriate way to do that is with testing in an approved lab, followed by genetic counseling. Because having the result is not always straightforward. The genetic variants that I talked about do not necessarily result in disease. So the penetrance, in other words, the expression of disease in people who carry the variant, may vary quite a bit. It may vary depending on age. It may vary depending on other genetic modifiers, most of which have not yet been identified. And it may vary based on environmental factors. So this is not a straightforward thing of saying you've got an abnormal blood test, so you're going to get Parkinson's. And people need to understand what the implications are before they have the testing rather than afterwards. So we really favor doing this, if at all, through a genetic counselor. Now, first of all, there aren't enough genetic counselors in the healthcare system, but we can occasionally refer people for that. The other way of doing the testing is through research. And I think you're probably all familiar with the Canadian Open Parkinson Network, C-Open. There is a way to to you can either register if you attend the clinic, you can be registered in person on C-Open and go through the testing, but you can actually do this online. And if you just look up Canadian Open Parkinson Network, you'll be taken to the links where you can register yourself. And as part of that study, DNA samples are being taken and sent to Montreal, where a geneticist with a lot of expertise in Parkinson's is assessing them. And they're incidentally being shared with an international consortium of genetic studies in Parkinson's known as G2P2. Is the gut involved? Yes, but it's complicated because there have been a lot of studies done. The question has been, does Parkinson's start in the gut, that something happens and then you get an inflammatory response, which includes misfolding of alpha-synuclein, and that triggers a cascade that travels up to the brain. One of the problems of interpreting the results of the microbiome studies, the studies that look at the expression of different bacteria in the gut, is that you don't know which is the chicken and which is the egg. So is it because of abnormal bacteria that people develop Parkinson's, or is it because the Parkinson's causes constipation and that leads to changes in the gut? Is there an association between RBD and cognition? Short answer is yes. It's thought that people whose Parkinson's starts with RBD may be more prone to cognitive change, but this is, again, I don't personally like categorizing people. There is a lot of literature on categorization of Parkinson's. And the fact is, I've, over my career, seen thousands of people with Parkinson's disease, and I would say no two are exactly the same. So I don't think we're at a stage where we can necessarily predict trajectory yet. And then a question on, am I familiar with the work of Alberto Espe? Yes, he's a good friend. I would say he's a very smart guy, but he stands in the minority on his particular perspective. In fact, the target engagement slide that I showed you earlier on Alzheimer's disease was one of Alberto's, taken from one of Alberto's papers. In that particular study, he was making the point that drugs that leach out amyloid don't necessarily lead to benefit in Alzheimer's disease. He's making exactly the same argument for Parkinson's disease, saying that the problem is not misfolded alpha-synuclein, but rather the lack of normal alpha-synuclein. He's right that alpha-synuclein has a normal function, which I pointed out is in the nerve terminal because it's thought to be important for the dynamics of neurotransmitter release, amongst other things. But I don't think most people would agree with him that misfolding of alpha-synuclein has nothing to do with Parkinson's. There's a whole line of evidence to support it. First of all, people with genetic mutations where they express too much alpha-syn are going to be highly prone to Parkinson's. And then there are other reasons based on experimental studies. Wow, we're getting a lot of questions here. Can your regular movement disorder neurologist distinguish between PD, PSP, CD, and RBD? There's no such thing as a regular movement disorder neurologist. We're all brilliant and in a category of our own. I thought that should be clear, but yeah, I mean, usually the answer is yes, but it's not always easy, even in expert hands. And the likelihood of being accurate in that differentiation is much greater the longer you follow the person. So if you follow somebody for 5 years, you're going to stand an 85% or better chance of making the correct diagnosis. But if you're seeing somebody very early on, it's much more difficult, and that's where imaging can be helpful. There's a question here about first Lewy body, second essential tremor. with meds, still trembling persists. I don't think I can answer an individual question here. A Lewy body disorder is suggested by the presence of nightmares. But you can have both Parkinson's and essential tremor. Even if it's tremor due to Parkinson's, it doesn't always respond to Parkinson meds. Around 2/3 of people get a response. But if it's essential tremor, it would be different medications. So this is really something that has to be discussed with your neurologist. Whether or not more testing would be helpful, I suspect not so much. And I've been asked about a link to the genetic research site. I don't have it. I don't have a link to the genetic research site unless you're talking about G2P2. Let me see if I can find that. What I talked about was the Canadian Open Parkinson's Network. And you can just look that up. Just look up Google Canadian Open Parkinson's Network. Of course, this is showing me reminding me that everything is based on perspective. G2P2 to a family doctor or an obstetrician means gravidum 2, para 2, which means you've been pregnant twice and you've delivered twice. To me, it means something different. So I'm just going to have to look a little bit harder to. I'm still not finding this. Oh, here we are. No. Here. Oh, it's sorry, GP2. I'll put that in the chat. And while I'm here, let me see if I can find the Canadian Open Parkinson's Network. And here it is. And then there's a question on, can you quantify the free water? Yes, you can. And that's what is being considered as a potential marker of disease progression, but it is not considered as prime time yet. Most of this work has been done by my colleague at the University of Florida, who is superb at this type of work. But this is not something that's routinely available, nor would there be, even in his hands, it's not clear how useful it's going to be as a marker, say for, if you want to conduct a trial of a disease-modifying drug. I would absolutely include it in conjunction with all the other stuff I've talked about, or not all of the other stuff, but a number of the other things that I've talked about, because I think it is worth pursuing. But it's not known yet whether it would be a robust marker of disease progression. And I just want to reemphasize that we have these markers that look as if they're quantitative. It's a lab test, so people feel more comfortable with that. But if it doesn't actually translate into better quality of life or a better outcome for the individual, who really cares? So, and I really mean that, because I think there's a huge gap between what is currently available as a biomarker and being able to measure the impact of a treatment that really makes a difference to people. Horse or cart. It's that PD leads to loss of smell. It's not that loss of smell leads to PD. However, loss of smell is, there are lots of reasons that you can lose your sense of smell, including things like allergy or having been in a fight. brain trauma is actually a fairly important cause of loss of the sense of smell. Those things don't necessarily lead to Parkinson's. It's that whatever is going on in Parkinson's um, is probably leading to loss of sense of smell that may become apparent before the motor signs. And in fact, the recent work done with the seeding amplification assay that I talked about towards the end suggests that if you do seeding amplification assays in people who have lost their sense of smell, they're positive far more often than I would have anticipated. I can't remember the exact numbers, but it's quite high, because other neurodegenerative disorders will also lead to the loss of sense of smell. Looks as if the questions have slowed down. I'm grateful for your interest. I'm really impressed by the quality of some of the questions as well. Any more, or are we going to call it a wrap?
I think that looks like all the questions that we have for today.
Okay, great. Well, I hope you
Uh,
found this somewhat informative, and thank you for your interest.
all right. Thank you, Dr. Stoessl.
Oh, there's
Oh,
another question. Anything to help RBD? Yeah, the commonest approach is to use melatonin, which is for most people a very well tolerated drug that you can get over the counter without a prescription. There are other treatments if melatonin doesn't work. A drug called gabapentin and a drug called clonazepam. But as in that order, you're more prone to get side effects. So we'll almost always start with melatonin. I'm not sure that I understand. Can you text for tau protein? What does that? I'm not sure I understand the question. Oh, test. Yes. So there's spinal fluid tests that can be done. We've done a lot of work with imaging. There are several tau imaging tracers out there. In fact, I showed you one, but I showed it to you because we thought it was labeling alpha-synuclein. And this is actually a problem. In our case, it was an advantage because we're very interested in studying Parkinson's. But most of the tau tracers that have been developed for PET have a big problem with off-target binding. In other words, they bind to stuff in addition to tau, and the correlation with the degree of tau pathology is not clear. This is actually an incredibly important question, not so much for Parkinson's, but for Alzheimer's, because the most recent drug that is not yet approved in Canada, to my knowledge, but the major paper that just appeared very recently in Alzheimer's was for another antibody called donanemab. And it appeared that, so to do this, you need to know that the person has amyloid pathology. So you could do a PET scan looking for amyloid, or you could do spinal fluid tests for amyloid. But it appeared in the donanemab study that the people who had low tau did better than the people with high tau. So it may well be that if you're wanting to give these drugs to people with Alzheimer's disease, you may need to test not only for amyloid, but also for tau. And then your follow-up is, how about PSP? Yes, well, we started that, and we've published, and lots of other people have published results on tau imaging in PSP. As a diagnostic test, I don't think most of us would want to depend on that, partly because of the off-target binding problem, but also because it's a very expensive and technically challenging way to test. With some of the advances in MRI, some of which I showed you, some of which I did not, that may be a better way to do that test. Question about gait. Well, Parkinson's is obviously associated with abnormal gait. That has been linked to abnormal cholinergic function, but this is a pretty complicated topic. What I would say is, in terms of people with freezing and or falling, which are two different problems, one of the things you want to consider is, are these problems occurring when the person is off? In other words, is it responsive to levodopa? If the answer is yes or maybe, then you would try to adjust medication. If the answer is definitely no, then first of all, most surgeons would be very hesitant to operate, to do deep brain stimulation. But the other thing is that the approach to managing it would probably be based on non-pharmacological approaches, at least in part, because gait and posture are cognitive functions. Normally, we take for granted our ability to stand up, walk, and do something else at the same time, like talk to somebody, watch the news as you're walking past, or watch a game that's on. You need a lot of cognitive control. And so if you're dual tasking, which means if you're paying attention to anything else, your chances of freezing or falling increase dramatically. So a lot of the management of the problem has to do with re-emphasizing for people that when you're up on your feet, your only job is to stay upright. Somebody's asking if you've had PD for most of your life. Is this possible? Well, it depends how you define PD. And what I didn't get into is with the recent seeding amplification assays, there are now a number of views. I can tell you, I'm an author on one of the papers that is going to be appearing in Lancet Neurology, we think at the beginning of the new year. The PPMI group, whose paper I showed you, I believe have another somewhat different definition coming out. And we have somewhat different views of this. So the group that I've worked with includes Dr. Lang in Toronto, people in Germany, elsewhere in Canada, in the US. We have laid out what we would propose as specific biological criteria for the diagnosis of Parkinson's, but for research purposes. The PPMI group, I think, would basically say that if you have an abnormal seeding aggregation assay, I don't want to oversimplify their perspective, but I think they'd say if you have an abnormal seeding amplification assay, you've got Parkinson's. But as I already indicated, you could have an abnormal test and not develop Parkinson's for many years or never develop Parkinson's. And there are many other reasons that I think that perspective is an oversimplification. They've linked theirs to a staging system, and I personally don't think we understand enough about the biology of Parkinson's to be able to say that there's one trajectory through which everybody passes. And then, could imaging help prove or disprove? I'm not sure how imaging would necessarily do that. I am familiar with his hypothesis and spent a long time talking to him a few months ago. I simply don't buy it. I mean, what his hypothesis was, what he really wanted to do was give a drug that would actually deplete dopamine from the neurons. And I think that will only make people worse. It's not, what you're talking about isn't completely crazy, though, the way the imaging would work. So there's been a long standing suggestion that dopamine is toxic. And that's why so it's packaged in vesicles, which is what I showed you at the very beginning. We can actually study the packaging process. So theoretically, if something goes wrong with the packaging and then you have loose dopamine floating around, it could be toxic. And that may well be the case. But the corollary of that is not true. So what's been suggested for a long time is that if you take levodopa, it may treat the symptoms, but it actually hastens the disease because it gets converted to dopamine and that causes damage. There is absolutely no evidence to support that. And if anything, the evidence goes to the contrary, in that levodopa may, in a brain, as opposed to in a test tube in isolation, may actually have growth promoting properties, but more importantly, treating the symptoms adequately may make the difference between getting enough exercise and not getting enough exercise. And so if a little bit of levodopa keeps you active, that is a far more important thing to do. Okay, I think I'll consider that a wrap. And thank you for your clinical trials on stem cell therapy. Yes, and I've actually been asked to be an advisor on some of those. I'm not going to say anything much about it. There are advances in stem cells. It's still not ready for prime time, and when you think about it, it's a bit of an oversimplification because we lose more than dopamine in the brain. So the stem cells are a good way of replacing dopamine, but we're not bad at doing that with medications. What you need is something to deal with all the other problems. Okay, thank you all very much for your interest and attention. Take care.
Thank you, Dr. Stoessl. Bye. Have a good weekend.
Bye.